This method can be used to dissect the cellular and molecular mechanism of FcγRIIIa-driven events without the need for target cells that express the antigen.
Set a refrigerated microcentrifuge to 4 degrees Celsius or place a microcentrifuge into a refrigerated space. Dispense 100 microliters of resuspended natural killer cells into 1.5 milliliter tubes or a 96-well U-bottom plate, and place the cells on ice. Prepare rituximab or other antibody of interest at a concentration of 100 micrograms per milliliter. Add 1 microliter to each tube of cells for a final concentration of 1 microgram per milliliter. And incubate the cells on ice for 30 minutes. During incubation, prepare a solution of anti-human kappa light chain antibody in media at a concentration of 50 micrograms per milliliter. The amount of antibody solution needed will be 50 microliters per cell sample. Warm the antibody solution to 37 degrees Celsius on a heat block or in a water bath. When incubation of the cells is complete, add 1 milliliter of ice-cold media to each tube, and centrifuge the cells at 135 times G and 4 degrees Celsius for 5 minutes.